Hi everyone, this is Ms. Romani, and for this lesson we will be learning about the process by which DNA makes a copy of itself, DNA replication. And as always, we will start by looking at the big picture. In grade 11, you learned about mitosis and meiosis, and I hope that you remember that prior to mitosis or meiosis, there was a stage called interphase. And during interphase, the cell gets ready for cell division, and one of the main things that has to happen during interphase happens during the S phase of interphase, and that is where the DNA gets copied. And that's extremely important because if a cell needs to make a copy of itself, well, first needs to copy its DNA so that each new daughter cell has a full complement of the original DNA strand. And so let's imagine now that it's 1953 and the double helix structure of DNA has just been discovered. So now scientists knew that DNA consisted of two separate strands that were bound to each other by hydrogen bonds. One big question that scientists asked themselves right after the discovery of the structure of DNA concerned DNA replication. Once you know the structure of DNA, that really provides a big hint as to how copying might take place. So it seemed very likely that the two complementary strands of the helix might actually separate during replication, each serving as what we call a template for the construction of a new and matching strand. So there were three basic models for DNA replication that had been proposed by the scientific community. The first one is called conservative replication. And in this model, DNA replication basically results in one molecule that consists of both original DNA strands, which we would call the parent strands of DNA, and another molecule that consists of two new strands that would be exactly the same as the parent strand, and we would call these the daughter strands of DNA. Now, another proposed hypothesis would be semiconservative replication. Now, during semiconservative replication, the two strands of DNA would unwind, and each of them would act as a template for the synthesis of a new and complementary strands, resulting in two molecules with one original parent strand and one new daughter strand. A third model was proposed. It was called dispersive replication. And in this dispersive model, DNA replication actually results in two DNA molecules that are mixtures or hybrids of both parental and daughter DNA. In this model, each individual strand is basically a patchwork of both original and new DNA. Experimentation was needed. So this leads us perhaps to one of the most ingenious experiments in biochemistry, the Mesosolahl Experiment. It was conducted by two scientists, Matt Meselson and Franklin Stahl, who originally met in the summer of 1954, just a year after Watson and Crick published their paper on the structure of DNA. And so they decided to team up and try to solve the puzzle of DNA replication. So they decided to work with E. coli bacteria. And they cultured the E. coli bacteria in a medium that contained a very heavy isotope of nitrogen called nitrogen 15. What that means is that whenever the bacteria were trying to make their new DNA strands, as bacteria do when they replicate, they would pick up nitrogen in the broth in which they were grown that was particularly heavy. And so by the end of a few cycles of growth, all the bacteria in this broth that they had their DNA, made up of heavy nitrogen. So it had heavy DNA because all the nitrogen in the DNA would be made up of this heavy isotope. So that's the beginning of the setup of the experiment. Then what they did is they transferred some of the bacteria culture into a second flask that contained regular nitrogen. This new nitrogen, this regular nitrogen, was a lighter nitrogen, nitrogen 14. And so they let the bacteria sit there for 20 minutes, which allowed for one cycle of replication because E. coli replicate every 20 minutes. So the bacteria would be allowed to replicate once in this experiment. And so the idea was that if you take a look at the DNA sample after 20 minutes, after one single replication, and you take a sample of that and you centrifuge it, that is, you put it in a machine that spins test tubes with the DNA really, really fast and allows the DNA to separate by weight, then you would be able to see where the heavy nitrogen is and where the lighter nitrogen is. So the idea was that by using this particular process, you would be able to figure out whether or not the DNA was replicating conservatively, semi-conservatively, or dispersively. If you have conservative replication, when the samples were centrifuged, you would end up with two lines in that sample. One that contained the heavy DNA or the parent DNA, and one that contained the lighter DNA or the daughter DNA. If the DNA was replicated semi-conservatively, you would end up with just a single line in the centrifuge because all the DNA strands would be a complement of both the parent and the daughter DNA. And as a matter of fact, the dispersive model would also end up with a similar line in the middle. So a second replication would be needed to really figure out if there's a difference between the semi-conservative and dispersive. Because if you think about it, if you let the bacteria replicate for another round, another 20 minutes, and you centrifuge it again, if the replication was conservative, you would end up with one bottom line with the original parent DNA, but each of the new daughter strands would have now replicated themselves, and you would end up with a much thicker line of lighter DNA. If it's semi-conservative, you would end up with a very light bar at the top where the brand new DNA was built, but you would also have a middle line with the DNA that contained the original parent strand and the new strand. And then if it was dispersive, you would end up with just one really thick line in the middle, maybe a little bit higher up as new nitrogen is being added, but you would not have the two separate lines because you would not have one DNA strand or a couple of DNA strands that were made up with both the original heavy nitrogen parent strand and the lighter nitrogen daughter strand. After the first replication, the results showed a single bar in the middle. So right away, they could eliminate conservative replication as a possibility. And both semi-conservative and dispersive were possible solutions or possible conclusions to the experiment. So another experiment was needed, and that's when they did the second replication. And after the second replication, they got results that basically proved that it was not conservative, obviously, but it was also not dispersive. And then it was actually semi-conservative replication. And that's the experiment. And so they figured out that DNA copies itself in a semi-conservative way, where the parent strand actually ends up becoming a template to the two new daughter strands. So let's take a look at exactly how this semi-conservative replication happens. And it's a very simple process. Essentially, you start with a parent strand of DNA. The parent strand needs to be separated into two separate strands. The two parent strands serve as a template, and the new strands are built using the template strand by using the idea of complementary base pairing. Wherever there's an adenine, a thymine would be added. Whenever there's a cytosine, a guanine. Wherever there's a thymine, an adenine. And whenever there's a guanine, a cytosine. So the complementary base pairs essentially allow each parent DNA strand to serve as a template for the new DNA strand. And that's it. That's it for the big picture, at least. Obviously, there's a little bit more to this, and so let's take a look at the details. And before we begin, let's look at a very important concept in the structure of DNA, and that is directionality of DNA. The idea that DNA has a 5' end and what we call a 3' end. And so this all brings us back to biochemistry, and we're taking a look at sugars. And in the unit on sugars, we talked about how we can actually number the carbons in a sugar molecule. Well, DNA is made up of nucleotides, and each nucleotide has a sugar, a deoxyribose sugar to be precise. The deoxyribose sugar has 5 carbons, so we name this carbon, starting with the 1' carbon, which is the carbon that is attached to the nitrogenous base in the molecule, then the 2' carbon, the 3' carbon, the 4' carbon, and the 5' carbon. The 5' carbon is the one that is attached to the phosphate group. So if we take a look at the molecule, then the bottom of the molecule would be the 3' end of the molecule, and the top of the molecule would be the 5' end, which is the end with the phosphate group is, because the phosphate group is attached to the 5' carbon. And so when the molecules of DNA are put together, the DNA strands are actually not equal to each other as they're placed together. They're what we call anti-parallel to each other. So whereas one strand of DNA would run in the 5' to 3' direction, the other strand, its complementary strand, would run anti-parallel in the opposite 3' to 5' direction. I always like to kind of picture it almost like if you have a person that is lying down and another person lying down right next to them, it would be like as if they were lying down head to toe. And so DNA replication depends on this directionality because DNA is always replicated in a single direction, and that direction is 5' to 3'. So the phosphodiester bonds that are formed are always formed by adding to the 3' end of the molecule and basically building the new molecule, the new strand of DNA, one nucleotide at a time on a 5' to 3' direction. So if you take a look at the diagram on the right, where I have that arrow pointing down from 5 to 3, imagine if we keep adding nucleotides to the bottom of that DNA strand, to the 3' end. That's how DNA is built, by adding new nucleotides to the 3' end of the molecule and building the DNA on a 5' to 3' direction. Okay, so the next thing that we're going to take a look at before we take a look at exactly how DNA is built, let's talk about how we start building it. How do we start the separation of DNA? Because in order to start building DNA, you need to separate the parent strands. And it would be so easy to imagine that DNA separates the same way that you would separate the two strands of a zipper. Like imagine if you have a hoodie and it's zipped up and you wanted to separate the two strands and unzip the hoodie. You would start at one end and you would zip it down. Unfortunately, that's not how DNA is separated. DNA is actually separated in what we call bubbles. Essentially, throughout the zipper, it is opened up right in the middle in many different places. Imagine how annoying that would be. If somebody, instead of unzipping their hoodie from top to bottom, they would just come around and then just separate it in several different places throughout the belly of the hoodie. And so that's essentially what happens. So there's always going to be something called a replication origin or origin of replication, where the DNA replication begins, where the DNA, basically the parent strands, get separated. And there may be several, and there's usually several at the same time. So there's not just one place where it starts in several places at the same time, forming a lot of these replication origins that have something called a replication fork, which is where the strands are still joined, forming what we call replication bubbles, which essentially look like bubbles when you look at it under the microscope, but eventually, as the DNA continues to separate throughout the strand and the new strands of DNA get built, the replication bubbles will eventually merge together, forming two daughter DNA molecules. So we're not starting at one end, moving towards the other end. We're starting in several places throughout the DNA strand, opening up the DNA strand in these tiny little bubbles. And then there's the replication forks, which is where the DNA strands are still joined. And basically the new DNA strands are going to be built starting from the origin of replication, where the bubble was first started, where the DNA was first split and separated, moving towards either end of the replication forks, towards the right and towards the left at the same time, until basically all the replication bubbles become two separate DNA molecules and they merge together. Let's go to that origin of replication. How do we start separating DNA? And that's the first step, the separation of the DNA strands. And it all starts with a molecule called helicase. Helicase is an enzyme whose job is essentially to unwind the double helix by breaking the hydrogen bonds between the bases. So we start there with the unwinding of the double helix by helicase. Now, one of the things that is going to happen though, as soon as the helicase moves through the DNA strand and winding it, is that the DNA strand is going to want to reform those hydrogen bonds because the adenines are attracted to the thymines, the guanines are attracted to the cytosines, and so they're going to want to reanneal, rebond. And so to prevent that and allow time for the new DNA strands to form, a new molecule is added. They're called single strand binding proteins. And it's not just a single molecule, it's a series of tiny little proteins that attach to both the parent strands. And what they do is they just prevent the reannealing of the parent strands, allowing time for the new DNA molecules to be synthesized. One of the things that it's going to be needed though is as DNA helicase moves through the DNA strand, unwinding it, is you're going to need to have a way of relieving the tension that is going to be building as DNA basically unwinds the two parent strands. And the reason for that is because DNA is not just a ladder, it's a twisted ladder. It's a double helix. And so imagine if you have a rope and it has a braid, it's braided, and you start to unravel the rope somewhere in the middle. Whilst you move through the rope and you start to keep unraveling it, you're going to build tension in either end of the rope as the helix of the rope just gets tighter and tighter. And so that can actually cause DNA to break. So in order to relieve that tension and prevent DNA from being damaged, a second molecule, a second enzyme called topoisomerase comes in. And its job is just to sort of move behind DNA helicase and cut the strands of DNA, allowing them to unwind a little bit, relieving that tension, and then resealing the cuts because we don't want the DNA to be cut up into pieces. We just want to relieve the tension that is formed as helicase moves through the DNA strand. Okay, so let's talk about how the strands are going to be built then. It's a process called elongation, and we're actually going to look at elongation in two stages. We're going to look at one strand being built. We're going to call that the leading strand, and then we're going to take a look at a second strand, and we're going to call that the lagging strand. And we call the leading strand because it is built from 5' to 3' without interruption. And so we'll start there because that's the easiest place to start. The molecule that is essentially in charge of building the new DNA strand, the star molecule here, is an enzyme called DNA polymerase 3. And DNA polymerase 3 builds the new DNA strand in the 5' to 3' direction, and it just attaches to the parent strand, and its job is to just bring the appropriate complementary bases to the bases that it sees in the DNA in the parent strand. So if it sees an adenine, it brings in a thymine, and so on. Now, the way that DNA polymerase works is that, like I said before, it builds it in the 5' to 3' direction. So here's the 5' direction, which is the 5' carbon that is attached to the phosphate group, and here's the 3' carbon. So it's basically going to be adding DNA, new nucleotides, to the 3' end, and the new strand is going to be built 5' to 3' that way. Here is a way of looking at it. Right here in this picture, you can see how you have the template strand in the darker blue, and you have the new strand that is being built, and the nucleotides are being added to the 3' end of the molecule, essentially building DNA from 5' to 3'. Now you might notice something a little bit strange about that nucleotide that is being added, that thymine that is being added to that new strand of DNA, and is that it has 3 phosphate groups. And if you remember anything about DNA, you remember that the nucleotides only have a single phosphate group, right? ATP has 3 phosphate groups, but not the nucleotides of DNA or RNA. But that is not actually the case. The reality is that there are actually 3 phosphate groups, in the nucleotides, because this is where the energy comes from for replication. So it's a pretty neat thing that ATP is not the only molecule that provides energy to the cell. As a matter of fact, when DNA is built, each of the nucleotides brings in its own phosphate groups for energy. So when adenines, thymines, guanines, and cytosines get added, they don't have a single phosphate group. They actually have 3 phosphate groups. And in this particular case, the bonds between the first and second phosphate group are broken, and the energy that is released by breaking these high energy bonds is used for the synthesis of the new DNA strand. So we don't have to use ATP energy to build DNA. The adenines, guanines, cytosines, and thymines come with their energy. As you can see here, you have the parent strand. Here's a new cytosine coming in. It has 3 phosphate groups. There's going to be a hydrolysis reaction. It's going to remove 2 of those phosphate groups, releasing energy. Notice that the new nucleotides are being added to the 3' end of the sugar, building that bond on a 5' to 3' direction. So DNA polymerase 3 essentially cannot just start building DNA without something called a primer. It needs to have something to which to add this new nucleotide to. And so the process of DNA replication actually starts with the addition of something called a primer. that is a short piece that is complementary to a part of the parent strand, and that primer is added by an enzyme called RNA primase. And as the name might suggest, RNA primase adds an RNA primer into the DNA molecule, into the template strand of DNA, and then the DNA polymerase 3 can then start building onto that RNA primer. So let's take a look at the lagging strand and how we elongate that strand. And the lagging strand is built in pieces. We call that discontinuous building of the lagging strand, or built discontinuously. But it's also built in the 5' to 3' direction. So stop for a minute and take a look at why that is. Take a look at the strand of DNA that you see in that picture. Look at the top where it says leading strand. The leading strand is that green piece of DNA that is being built. Follow that arrow. The arrow is moving from left to right. So that leading strand is moving continuously from left to right. It is being built from 5'. The 5' direction is where the primer is in that picture, is on the very, very left hand side. And then it's just being built all the way down, continuously following that arrow, 5' to 3'. Why is that arrow moving in that direction? That's the direction that helicase is moving. So helicase is basically unzipping the DNA, and DNA polymerase is following it along, and as more and more of the DNA is unwound, there it goes. It just continues to build a new DNA strand that way. That's the leading strand. It builds it in a continuous manner. But the lagging strand, it cannot follow helicase because it also has to build DNA in a 5' to 3' direction. And because the strands of DNA are anti-parallel to each other, the complementary strands are anti-parallel to each other, the parent strand will not allow the build of a new DNA strand on a 5' to 3' direction that is going to be moving in that left to right direction. And so that strand has to be built in pieces. And these pieces are called Okazaki fragments. Now, that obviously cannot be the end of the story because the leading strand is being built and it looks beautiful and it's intact, but the lagging strand is made up of fragments and pieces. And of course, you cannot have the DNA strand have all these little gaps between them. It has to be a continuous molecule. Not only that, but remember, we have all these primers there that are made out of RNA. And so we don't want RNA within our DNA. We want a continuous piece of DNA. So this is where DNA polymerase 1 comes in. It is a different enzyme than DNA polymerase 3. It is obviously similar to that. And DNA polymerase 1 has one very important job. It removes the RNA primers and replaces them with DNA. And then that way, now the Okazaki fragments will be made up entirely of DNA. There will be no more RNA primers. Once all the Okazaki fragments have had their primers replaced by DNA polymerase 1, then a new enzyme called DNA ligase can come in and essentially join the pieces of lagging strand together by creating phosphodiester bonds between those pieces, between those Okazaki fragments. And finally, it's the editing and proofreading of DNA. So once the two strands, the leading and the lagging strand, are finished being built, the DNA polymerase enzymes themselves, both DNA polymerase 1 and 3, act as exonucleases. Exonucleases are basically enzymes that fix any errors in DNA replication. They read the code, they find any mismatches. If there is an adenine added instead of guanine, say for example, they just fix them and repair any mistakes that are made. And that's it for today's lesson. I highly recommend that you follow up this lesson video by watching some of the animation videos that I've linked on the classroom website. A continuous animation of the process is going to be helpful to better understanding this process. Have a great day. Talk to you soon.